This audio series is brought to you by PharmaState, an exclusive application for pharma industry professionals.
Hello friends, in this audio series you will listen to 48 most frequently asked interview questions for quality assurance profile in pharmaceutical industry. This audio series is in 4 parts. Each audio has 12 questions and answers. This is second part containing next 12 questions and answers. Question number 13. Explain few ICH stability guidelines. Answer: Q1A stability testing of new drug substance and products. Q1B photostability testing of new drug substance and products. Q1C stability testing of new dosage forms. Q1D bracketing and matrixing designs for testing of new drug substances and products. Q1E evaluation of stability data. Q1F stability data package for registration applications in climatic zone 3rd and 4th withdrawal. Question number 14. What is significant changes in stability testing? Answer: followings are significant chances in stability testing. 1. 5% change in assay for initial value. Second, any degradation product exceeds its acceptance criterion. Third, failure to meet acceptance criterion for appearance, physical attributes and functionality test. 4. Failure to meet acceptance criteria for dissolution for 12 units. Question number 15. What needs to be checked during in-process QA checks? Answer: following checks are done. A. Environmental monitoring. B. Measured value obtaining from the process equipment, temperature, RPM, etc. C. Measured value obtained from person, timing, entries, etc. D. Process attributes, example, weight, hardness, friability, etc. Question number 16. What precautions shall be taken while collecting in-process samples? Answer: while collecting in-process samples, avoid contamination of the product being sampled. Means don't collect samples with bare hands and avoid contamination of sample taken. Question number 17. In a tablet manufacturing facility, why positive pressure is maintained in processing area or service corridors? Answer: in tablet manufacturing facilities, pressure gradients are maintained to avoid cross-contamination of products through air. Usually, processing area are maintained under positive pressure with respect to service corridors. Question number 18. What is the purpose of stress testing in stability studies? Answer: stress testing of the drug substance can help identify the likely degradation product, which can in turn help establish the degradation pathway and the intrinsic stability of the molecule and validate the stability, indicating power of the analytical procedure used. The nature of the stress testing will depend on the individual drug substance and the type of drug product involved. Question number 19. What is dead leg? Answer. A dead leg is defined as an area in a piping system where liquid can become stagnant and not be exchanged during flushing. Question number 20. Are force degradation, stress testing and accelerated stability testing same? Answer. Force degradation and stress testing are not same. Stress testing is likely to be carried out on a single batch of the drug substance. The testing should include the effect of temperature in 10 degrees Celsius increments, example 50 degrees Celsius to 60 degrees Celsius above that the accelerated testing. Humidity, example 75% relative humidity or greater. Where appropriate oxidation and photolysis on the drug substance, the testing should also evaluate the susceptibility of the drug substance to hydrolysis across a wide range of pH values when in solution or suspension. Photostability testing should be an integral part of stress testing. Question number 21. Why do we calibrate a qualified equipment instrument on definite intervals? Answer. An equipment or instrument can drift out of accuracy between the time of qualification and actual use. So, it is recommended to calibrate and recalibrate the measuring devices and instruments on predetermined time intervals to gain confidence on the accuracy of the data. Question number 22. Why do we consider 3 consecutive runs batches for process validation? Why not 2 or 4? Answer. The number of batches produced in the validation exercise should be sufficient to allow the normal extent of variation and trends to be established and to provide sufficient data for evaluation and reproducibility. First batch quality is accidental, coincidental. Second batch quality is regular, accidental. Third batch quality is validation, confirmation. In 2 batch, we cannot assure the reproducibility of data. 4 batches can be taken, but the time and cost are involved. Question number 23. Explain about revalidation criteria of AHU system. Answer. AHU system shall be revalidated periodically and mentioned in regulatory standards. AHU shall be revalidated in following cases also: when basic designs of AHU is changed. Second, when clean room volume is changed. Third, when new equipment is installed. And the last one is when a construction is carried out that calls for reconstruction of AHU system. Question number 24. What needs to be checked during AHU validation? During AHU validation, following tests shall be carried out. First, filter efficiency test. Second, air velocity and number of air changes. Third, airflow pattern. Fourth, differential pressure, temperature and RH. Relative humidity. Fifth, static condition area qualification. Sixth, dynamic condition qualification. Seventh, non-viable count. 8. Microbial monitoring. 9. Area recovery and power failure study. Thank you.